Welcome to my talk. As you heard in my pitch yesterday, I'm going to talk about specialized plant metabolites. And firstly, I would like to talk about why these plants produce those compounds. So the metabolites are often produced in flowers, leaves, and roots of the plants in response to abiotic stress factors, for example, or also to attract pollinators. And while they are not specifically produced for humans, many of them actually do have health benefits. And yeah, I'm going to present the 4 plants that I've already talked about in my pitch yesterday. But before I do that, I would like to introduce you to the workflow of our projects in our labs. So at the beginning of all of our projects, probably not surprising, is DNA extraction. And DNA extraction from plants can sometimes be difficult. So what we usually do is that we incubate the plant in darkness for a couple of days and then do non-destructive sampling of plant material, mostly leaves, which we then grind in liquid nitrogen and then start the DNA extraction protocol. Because we only want high-quality DNA samples with a low level of fragmentation and no contamination, we do quality control steps before and after the short fragment depletion. And then we do my favorite part, which is library preparation and sequencing. Then we continue with the computational analyses. So we do base calling, of course, and quality control of the base called reads to make sure that it has the quality that we want for our analyses. And then we start assembling. And to evaluate the assembly, there are many different tools that we like to use, and what kind of tools we are using in the end always depends on the research question we are focusing on. And once the assembly is completed, we do the structural annotation. And for our structural annotations, we also like to use RNA sequencing data if it is available, just to make sure that it's as complete as possible. And then for the functional annotation, it's kind of the same thing. So the tool we are using depends on the question we are asking. For example, we like to use KIPES3, the MIP annotator, or BHLH annotator, which were all developed in our lab or in cooperation with our lab. And then when we do phylogenetic analyses, afterwards we submit the data open source so that everybody can have access to the genome sequences that we have produced. And now I would like to start with the first plant, which is Digitalis purpurea, more commonly known as foxglove. It's a very pretty ornamental plant, but also highly poisonous, so I would not recommend it touching or eating it. And it also has very pretty flower pigmentation in white or purple, which you can see in the pictures. And in the beginning, when we started working with this plant, we were interested in how the pigmentation loss occurs in the white flowers, and we have also uploaded a preprint on this if you're interested. And then we also had a look at the medicinal relevance, because Digitalis plants produce glycosides that are cardioactive, which means they can increase the cardiac output and used to be prescribed to patients suffering from heart conditions. And usually those compounds are isolated from a related species, Digitalis lanata, and we wanted to see if the synthesis of these compounds could also potentially happen in the Digitalis purpurea plants that we have in our institute's garden. So on the basis of a Digitalis lanata dataset, we were able to identify several enzymes and cytochromes that are involved in the process of digoxin production. You can see them indicated with the purple arrows, and underneath there are the gene IDs from our dataset, starting with a G and then the number. And you can see that we were able to find several enzymes that are involved in this pathway. And it seems as if those genes are pretty far away from each other on the genome. So we are assuming that there's not a biosynthetic gene cluster present here. And then to the next plant, which is Tropalium majus. It belongs to the Brassicales, which is a group of plants that produce glucosinolates. And glucosinolates are precursors of antimicrobial, antiviral, and also anti-carcinogenic compounds. And it is very well described in Arabidopsis, but not much is known about the glucosinolate biosynthesis in Trophalia majus. So what we do know is that there are 3 steps. And the first step is the elongation of side chains of precursor amino acids, and then the glucosinolate core structure is synthesized, and then in the end there are some secondary modifications taking place. And we were able to identify several enzymes, mostly transferases, in all 3 steps of the biosynthesis. So we are assuming that there is a certain level of conservation in the process of glucosinolate production, but this is just really a few enzymes of the whole pathway. The whole pathway is very complex, and now we are working towards identifying more genes that play a part in the production process. And next I have a plant called Urtica dioica, which many of you will know as stinging nettle. And the stinging nettle was sequenced in our data literacy in genome research class, supervised by Katharina. And Katharina actually has an online poster here at London Calling. It's poster number 63, if I'm not mistaken. So if you want to learn how we taught students about nanopore sequencing and bioinformatics, then check this out. And this plant has been used for medicinal purposes for a long time, especially to relieve joint pains and also as a diuretic. And Katharina was interested in routine biosynthesis, but I am personally also interested in viruses, and I have read that other teas, like green teas, can be given to mice, for example, and then protect them from severe influenza A disease. So I wanted to see if this could also be possible with stinging nettle tea. So we had a look at the proanthocyanidin biosynthesis, because catechin and epicatechins that are produced in this synthesis pathway have been described to have antiviral properties. And we were able to identify many genes in the pathway of those compounds, as well as genes encoding transcription factors that play a role in this pathway. So we are not going to test this on mice, but I'm definitely going to keep in mind when the next flu season is coming around that I might have a cup of stinging nettle tea or 2. And then the last plant I'm going to talk about today is Withania somnifera. And Withania somnifera belongs to a group of plants that produce withanolides. And withanolides are potentially used for cancer therapeutics because they have shown to have anti-proliferate activity on tumor cells. However, so far only the first step of withanolide biosynthesis is known, so it is our goal to work towards the unraveling of the whole biosynthesis pathway. And to do this, we sequenced the plant, did an annotated assembly, and then did synteny analyses with other withanolide producing plants. And you can see the withanolide producers here indicated in green and non-producers indicated in gray. And then what's obvious on first glance is that apparently we have identified a biosynthetic gene cluster that is like the borders of the gene clusters are indicated with the bold black lines, and you can see that there are many genes in the gene cluster that are not found in the non-producers. Additionally, we found that the gene responsible for the first step in withanolide biosynthesis is duplicated, which is also shown in all of the withanolide producers. Our Withania somnifera assembly used for this synteny plot was a preliminary assembly, but in our most recent assembly we have also found the duplication. And yeah, with this we are going to move forward and see that we can identify more genes of this biosynthesis pathway to work towards the unraveling of this pathway as well. And with this, I would like to acknowledge the amazing team at FOCA Lab at TU Braunschweig and our collaboration partners and funding agencies. And if there are any questions that can't be answered today, then please don't hesitate to get in touch per mail, for example. Thank you very much for your attention.